was a German scientist and working on this in the 1930s. He was Jewish and had to escape Germany before the war started and went to England. And there he continued to work, and he got the Nobel Prize for discovery of the citric acid cycle. He also discovered another pathway we're going to talk about this semester, the urea cycle that's involved in metabolism of amino acids. So he was a very productive scientist who made a lot of good contributions. Okay, so the glycolysis happens in what compartment of the cell? The cytosol, that's right. And this next steps we're going to talk about happen in mitochondria. So pyruvate has to get into mitochondria, and then we're going to talk about what happens with pyruvate. Pyruvate is decarboxylated. One carbon dioxide comes off by an enzyme complex called the pyruvate dehydrogenase complex. And the remaining 2 carbons are attached to coenzyme A. So you learned about coenzyme A in the introduction to metabolism with Dr. Zolkuska. Coenzyme A is a cofactor that's involved in moving acyl units around. In this case, it's a 2-carbon acyl unit. We'll see examples later on where we have longer carbon chains that are moved by coenzyme A. So these 2 carbons out at the end are the 2 carbons that came from pyruvate, and we'll see how they get there by the pyruvate dehydrogenase complex. So here's the overall reaction that occurs with the pyruvate dehydrogenase complex. It's a complex of enzymes, 3 different kinds of proteins I'll show you in a minute. So from glucose, we go through glycolysis and get 2 molecules of pyruvate. The pyruvate dehydrogenase complex removes the carbon dioxide and recovers some electrons from that pyruvate, and the product is acetyl coenzyme A. Acetyl-CoA then can enter the citric acid cycle to be completely oxidized to carbon dioxide and recover all the energy that's in that acetyl-CoA. Here's the reaction. So here's pyruvate, the structure you already know. Coenzyme A is used to transfer that 2-carbon unit to make acetyl-CoA. At the same time, there's a reduction of NAD to NADH accompanied by oxidation of the molecule to get acetyl-CoA. There are 3 enzymes, 3 kinds of proteins that make up the pyruvate dehydrogenase complex, and it's in a very big multi-protein complex. So with quaternary structure, in bacteria and E. coli, there are 24 copies of an enzyme called pyruvate dehydrogenase that we can talk about easier as enzyme 1. 24 copies of an enzyme dihydrolipoyl transacetylase that we'll call enzyme 2, and 12 copies of dihydrolipoyl dehydrogenase that we'll call enzyme 3. So it's a huge complex, and we can even see it with an electron microscope. So these particles are multiple proteins assembled into a complex for carrying out this reaction, removing 1 CO2 from pyruvate to make acetyl-CoA. A lot of what we know about this enzyme complex was done in our biochemistry department here by Tom Roach, who was a longtime member of our department, and his whole career was on studying how pyruvate dehydrogenase works and how it's regulated. So things that are in the textbooks about pyruvate dehydrogenase, a lot of it came from here. You learned about enzyme cofactors. So enzyme cofactors are small molecules that are not part of proteins, but may be attached to proteins and take part in the chemistry. And in the pyruvate dehydrogenase complex, there are 4 cofactors that we're going to look at. 2 of them you learned about already with Dr. Zolkuska, FAD and NAD. Those are involved in oxidation reduction reactions, and we'll look at those. There are two others that you probably haven't learned about yet. One is thiamine pyrophosphate. So here's thiamine pyrophosphate. This comes from a B vitamin, so you have to get thiamine in your diet from a vitamin. You can't make, your own cells can't make this molecule. The other one is lipoic acid. And lipoic acid is an 8-carbon carboxylic acid with sulfurs on carbons 6 and 8 that form a disulfide bond. And this disulfide bond can open and close, and that's part of the chemistry we're going to talk about. The part of the thiamine pyrophosphate that's involved in the chemistry is this carbon right here in the 5-membered ring between a nitrogen and a sulfur. So we're going to see that the acetyl-CoA is going to be attached there. Okay, so we're going to go through the reactions of the pyruvate dehydrogenase complex. Here's the first reaction. So here's that ring in thiamine pyrophosphate that I showed you a minute ago. And this carbon here is where the chemistry is going to occur. This enzyme E1, pyruvate dehydrogenase, catalyzes a decarboxylation, and so this carbon dioxide in pyruvate is released as carbon dioxide gas, and the remaining 2 carbons are transferred to this carbon on thiamine pyrophosphate. So this is hydroxyethyl thiamine pyrophosphate. This is a reduction reaction. This carbonyl in pyruvate is reduced to an alcohol. And so the product of the first step is this hydroxyethyl thiamine pyrophosphate. Next, we're going to see that the 2-carbon unit is going to be transferred to this lipoic acid. It's a cofactor in enzyme 2, and in enzyme 2, it's attached, so remember I told you it's a carboxylic acid, it's attached to the nitrogen in the side chain of a lysine residue in the enzyme 2. So here's the polypeptide of enzyme 2. It has a lysine in it, and it's a specific lysine in the sequence. And this carboxyl group forms an amide bond with the amine in the nitrogen at the end of the lysine side chain. And now we have this organic group hanging off of enzyme 2 with this disulfide bond. So that's involved in the second reaction. So this hydroxyethyl group here is transferred to the lipoamide in enzyme 2. And so there's a reduction of the disulfide bond and transfer of this carbon onto the sulfur. So this is now a thiol ester because we have a carboxylic acid here. So this group, this hydroxyl group, was oxidized to an acid, and then that acid can form a thiol ester with the sulfur in the lipoamide. So now we've transferred the 2-carbon unit that came from pyruvate to enzyme 2 in the complex through forming this bond with the lipoyl group. In the third reaction, that acetyl group is then transferred to coenzyme A. So that's the important product of this reaction. There's this 2-carbon unit now is forming a new thiol ester with the sulfur at the end of coenzyme A. So remember that coenzyme A has a sulfhydryl group at its end, and that's where it accepts the carboxylic acid. So now we have acetyl-CoA as one of the products, and the dihydrolipoamide is in this form with 2 free sulfhydryl groups. And the next step is to oxidize this to reform that disulfide bond. So that step is done using and FAD that's in enzyme 3. So in enzyme 3, there's an FAD cofactor. It accepts electrons to become reduced, and the dihydrolipoamide is oxidized to form a disulfide. So this cofactor is oxidized and the FAD is reduced to form FADH2. So that's another important product of the pyruvate dehydrogenase step. The next step is to transfer the electrons from FADH to NAD to make an NADH. So FADH2 is oxidized to FAD, NAD is reduced to NADH. The electrons are transferred from FADH to NAD. So that's the last step in the reaction pathway for the pyruvate dehydrogenase complex. We regenerate the FAD in enzyme 3. The lipoyl group attached to enzyme 2 functions as a kind of flexible arm. So it can grab that acetyl-CoA from enzyme 1 and move it over to enzyme 3. And in that way, the substrate isn't diffusing away from the active sites of the enzymes and has to diffuse back. It makes this enzyme much more efficient because the substrates and products can be moved around by the flexible organic and chemical that acts as an arm to move that molecule around. So this is a diagram of this pathway that we will go over it again looking at this. So here's the complex. It has enzyme 1, which is the pyruvate dehydrogenase, enzyme 2, and enzyme 3. And enzyme 2 has this lipoyl group attached. In the first step, the pyruvate is decarboxylated to form the thymine, the 2-carbon alcohol attached to thymine pyrophosphate. In the second step, now this lipoyl group swings up here and accepts that, and we form this carbon-sulfur bond, the thiol ester. And now we have this lipoyl group swings this down into carbon 2 and transfers that 2-carbon unit, the acetyl unit, to coenzyme A. So we get the acetyl-CoA product. Now the lipoyl group goes up to enzyme 3, where there's an FAD, and transfers electrons from the sulfhydryls to FAD to make FADH2 attached to enzyme 3. And the last step is transferring those electrons from FADH2 to NAD to get NADH. That's the other important product of this pathway. And now we're back at the starting state of the pyruvate dehydrogenase complex, ready to react with another molecule of pyruvate. We're going to see through the semester that pyruvate keeps turning up in different pathways. It's an important intermediate that is involved in lots of biochemistry, lots of metabolism. And in this pathway, we're starting with glucose making 2 pyruvates. The pyruvate dehydrogenase complex makes acetyl coenzyme A, and acetyl coenzyme A can go on to be completely oxidized to CO2 in the citric acid cycle. It can be used to synthesize fatty acids. It can be used for some other things. It can be used to synthesize some amino acids and to make cholesterol. So acetyl-CoA is another important molecule in metabolism. Because the pyruvate dehydrogenase is sort of central in this pathway, it has a lot of, it needs to be regulated. So cells are deciding how to use that pyruvate depending on the needs for energy or for building other molecules at that time. The pyruvate dehydrogenase is regulated by a kinase. So there's a specific pyruvate dehydrogenase kinase that phosphorylates that enzyme and inactivates it. So when a specific group in that enzyme is phosphorylated by a kinase. Now it's in an inactive form. There's a specific phosphatase that can cut off that phosphate to return it to the active form. So this is a fast way to regulate enzymes. It's kind of an on-off switch, and in a second you can turn the enzyme from on to off and back on again using these phosphorylation and dephosphorylation. We'll see that in other pathways in metabolism where kinases regulate the activity of a variety of different enzymes. Pyruvate dehydrogenase is also regulated by allosteric regulators, so small molecules that bind to the enzymes and change their activity. And so let's look at this, how this works. In a cell that has a high energy charge, that is, the ATP concentration is high relative to AMP and ADP, that cell doesn't need to make more ATP. And so it's going to inhibit the pyruvate dehydrogenase complex. And that's done by feedback inhibition. ATP is an inhibitor of the pyruvate dehydrogenase. NADH, which is a product of the reaction, inhibits pyruvate dehydrogenase, and acetyl-CoA, the main organic product of the pathway, inhibits it. So when the cell has a lot of energy built up, all of these things slow down pyruvate dehydrogenase complex. If the cell has a low energy charge, ATP concentrations are low, then ADP concentrations are high, and that activates pyruvate dehydrogenase. And high concentrations of pyruvate also activate pyruvate dehydrogenase. So this is a feed-forward activation. This is a feedback activation. So there's lots of regulatory mechanisms for pyruvate dehydrogenase. There's the phosphorylation reaction and these allosteric regulators that sense how much of acetyl-CoA the cell needs and speeds up or slows down the rate of the pyruvate dehydrogenase reaction. So this slide kind of states summarizes what I've been talking about. When ATP level is high, pyruvate dehydrogenase slows down because of allosteric inhibition by NADH and acetyl-CoA and activation of the kinase. So the kinase is activated, and that inactivates pyruvate dehydrogenase complex. When ATP is low, then we get relief of that inhibition, and pyruvate is converted to acetyl-CoA. So acetyl-CoA then can enter the citric acid cycle. That's the next pathway that's involved in getting all the energy out of a glucose molecule. Or we see later that this pathway is also involved in metabolizing the carbons that come from fatty acids. So in the citric acid cycle, acetyl-CoA is oxidized to 2 molecules of carbon dioxide. And the energy from those oxidation reactions is conserved by making NADH and FADH2. And there's also some production of GTP or ATP in this pathway. So the citric acid cycle is involved in getting the energy out of acetyl-CoA and conserving it to be used later by the cell. The enzymes for citric acid cycle are also in the mitochondrial matrix. So the matrix is the inside solution inside the mitochondria, and the pyruvate dehydrogenase complex is in the same compartment. So the acetyl-CoA that's formed from pyruvate is already in the right place in the cell, in the mitochondrial matrix, to be used by the enzymes of the citric acid cycle. So this cycle contains a number of enzymatic steps. The net reaction is that we start with an acetyl coenzyme A, a 2-carbon acetyl group. It's oxidized to 2 carbon dioxides. There are 3 NADs that are reduced to NADH, an FAD reduced to FADH2, a GDP that's phosphorylated to make a GTP, and the free coenzyme A is another product. So we're going to look at the steps for how that happens. This is a pathway that's a cycle. So we start with this molecule, oxaloacetate, and we go around the set of reactions and we end up with oxaloacetate again. We've seen oxaloacetate on Wednesday. What pathway was it in? Gluconeogenesis. So at the beginning of gluconeogenesis, oxaloacetate is formed. So there's a connection between gluconeogenesis and the citric acid cycle that we'll talk about some more. So you already know the structure of oxaloacetate. It's a 4-carbon compound. It combines with the 2-carbon acetyl group to form a 6-carbon compound citrate we'll look at. And as we go around this pathway, the important products are NADH and ATP, FADH2. And the carbons are oxidized to release as carbon dioxide. We're going to look at each individual step. The citric acid cycle is happening inside the mitochondrial matrix. So acetyl-CoA is oxidized to CO2, and the electrons are conserved as FADH2 and NADH, and those electrons are used in the electron transport chain in mitochondria as an energy source for making ATP, and we'll talk about that next week. So the electrons that came from acetyl-CoA are conserved as FADH2 and NADH, and important as an energy source for ATP synthesis. Acetyl-CoA can be made from glucose through glycolysis and pyruvate dehydrogenase complex. It can also be made by degradation of fatty acids and degradation of some amino acids. And we'll look at those later this semester, but all of them can result in formation of acetyl-CoA that's used as an energy source that's important for making ATP. So let's look at the reactions. The first reaction is citrate synthase. So here's oxaloacetate, the 4-carbon compound that we studied as one of the first steps in gluconeogenesis. It condenses with the 2 carbons from acetyl-CoA that to finally make citrate with release of coenzyme A. So this is citrate. It's a tricarboxylic acid. There are 3 carboxylic acid groups in this molecule. So that's why this pathway is sometimes called the TCA cycle, tricarboxylic acid cycle. So that's the first reaction, forming citrate from oxaloacetate and the acetyl group from acetyl-CoA. The second reaction is a rearrangement of citrate. It's moving that hydroxyl group up 1 carbon, so it's just a shifting. And it happens by a dehydration, so we remove a water molecule, forms a double bond, and then a rehydration, and adding that water across that double bond to have now the hydroxyl group at carbon 2. This molecule is called isocitrate. So it's just a simple kind of rearrangement, shifting a hydroxyl group from 1 carbon to another. The next step, it starts with isocitrate and is going to reduce NAD to NADH while oxidizing the isocitrate and removing a carbon dioxide. So the carbon dioxide comes from this CO2 in isocitrate. It's released as a carbon dioxide gas. And this carbon 2 is oxidized from an alcohol to a carbonyl. And so now we have a new 5-carbon molecule that is called alpha-ketobutyrate. So it has a carboxylic acid group at carbon 1, a ketone at carbon 2. And the electrons are conserved by reducing NAD to NADH. This is, we call this reaction an oxidative decarboxylation. So it's a decarboxylation because we're taking off that CO2, and it's an oxidation of this second carbon that results in reduction of NAD to NADH. This is an important regulatory step in the pathway, and we'll talk about that some more later today. You're going to see alpha-ketoglutarate again later in the semester, later in April, because alpha-ketoglutarate is important in amino acid synthesis. So we're going to see, especially for synthesis of glutamic acid, alpha-ketoglutarate is a precursor. The next step starts with alpha-ketoglutarate and is another oxidative decarboxylation. So this CO2 from this carboxyl group of alpha-ketoglutarate is removed as a CO2, and we have another reduction of an NADH to NAD. And this ketone now is converted to a carboxylic acid that's forming a thiol ester with a coenzyme A. So coenzyme A comes back into the picture again, forming a bond with this 4-carbon molecule now, succinyl-CoA. This is another oxidative decarboxylation and another important regulatory step in the citric acid cycle. Alpha-ketoglutarate dehydrogenase is interesting because it's very similar to pyruvate dehydrogenase. It's a complex of enzymes and carries out the same kind of reaction. So in pyruvate dehydrogenase, we saw decarboxylation and reduction of NAD to NADH. Alpha-ketoglutarate has the same cofactors. Alpha-ketoglutarate dehydrogenase has a lipoyl cofactor. It has a thiamine pyrophosphate cofactor. The mechanism of the reactions is very